Okay, in this lecture we will discuss about the different algorithms for aligning protein sequences. So, what did we discuss in the last lecture? You have different types of alignment, right? To have two sequences, what are the information you can directly get from these two sequences? Immediately what can we observe, right? One of the easiest one is dot plot. So, dot plot compares two sequences, right? If the sequence is the same, in amino acid is the same, then you put a dot. Then if you make a plot, you can observe with any matching sequences, right, exact matches, or you can see deletions or insertions in any of the two sequences. Then we discussed about the aligning sequences with some scores, right. Visually you can see the plot and look at the regions where you can see the similar residues or same residues, right. Then you can give some scores. For example, if here is a matching amino acid or matching nucleotide, we give a score, a reward, or if it is mismatching, we give penalty. Then we also introduce gap. So, what is the meaning of gaps in alignment? Insertions or deletions, right. So, in this case, comparing the sequences which have mutations or substitutions, the insertion and deletions are rare. So, we give penalty when we introduce a gap. So, now we have three different aspects, one is a match, mismatch and a gap. So, we give a reward for the match and we give a score for the mismatch and a penalty for the gaps. Then the gaps, we have two different types of gaps. What are two different types of gaps?
Gap of
Origination penalty plus gap penalty, like how many times gap is introduced and totally how many gaps, right. So, if you have different originations, so we have more penalties, right. So, we use these scores to align two sequences. Then we try to construct scoring matrices to align the sequences. So, different metrics for nucleic acids as well as for proteins. For the case of nucleic acids, right, how many bases? 4, right. So, in this case, based on the substitution, right, to either reward or the penalty, either it is mild penalty or severe penalty. For example, if we have purine to purine or pyrimidine to pyrimidine or other way, right, purine to pyrimidine or pyrimidine to purine. So, we give the penalty accordingly, so that we can give preference to match the similar sequences, right. So, when you look into proteins, there are various ways to align the protein sequences to give weightage to the amino acids. This is the one of the physical chemical properties, right. For example, if the two residues are charged, for example, aspartic acid and glutamic acid. So, in this case they are similar, right. This we give the positive score compared with aspartic acid is replaced by valine or any other hydrophobic residue. So, this is the pairing of these similar functional groups, right, either the aromatic groups or the non-polar and charged groups and so on. Either you give the score or you give the penalty. And we also compare the hydrophobicity or the charge as well as size. For example, if alanine and the serine, this is similar in size. So, if you compare the size, so they are similar, right. Accordingly, you can align the sequence, so you can give the score. Then also we discussed about the genetic code, how to give score based on genetic code, number of mutations in the DNA, then the codons, right, how many mutations in the codons. So, we from which type of mutations in the codons, then we can give weightage based on the genetic codes. Among all these things, how we derive the matrix, then finally we take the actual substitution rate. For example, if you get a set of sequences, get the sequences with the high sequence homology, from these sequence similarities we see what are the possible mutations. From that mutations, then we can derive the matrix, we can see what are the changes actually happen, right, and based on that we derive the matrices. So, this matrix is called the point accepted mutation matrix. For example, if you have 100 amino acid residues and 90 are same, right. So, how many variations? 10%, right, in this case 90% are similar. So, what are 10 variations? We get the information and then we see the substitutions, right, what are probable substitutions. So, here first we take in alignment, right, with the maybe at the high sequence identity, say more than 95%. Now, what are the various factors one has to consider to derive a PAM matrix? First we need to see, for example, A, how many alanines in the sequence, right, total number of residues in the sequence. how far alanine is mutating to other residues? What is the probability of mutating alanine to a specific residue, for example valine. So, here we show that for a relative mutability for each amino acid, how many times say alanine is mutated to others. And the second one, we need to see the exact mutations, for example Aij, how many times I is mutated to J. For example, we say ACM, number of time methionine is replaced with cysteine. Likewise, you can see the relative mutability of any amino acid. Then one has to consider the number of amino acids, right, and the total number of residues in the alignment. Then you take the log of these entries to get this PAM matrix, right. So, this is why we also call this PAM matrix as log odds matrix. We can derive the PAM matrix based on various sequence homologies. We can say 80%, we can say 90%, or we can say a very less homologous. So, if we do this, we use PAM1 matrix, that is to compare the sequences which are closely related, and we use PAM 1000 for comparing the sequences with the distance relationship, that means they are very not homologous to each other. So, usually in the literature we use PAM 250 for the sequence alignment, for example BLAST or FASTA use PAM 250 for the sequence alignment. So, I will show an example how to construct a PAM matrix. So, we see one sequence here A, C, G, C, T, A, F, K, I. In the first case A is mutated to G. So, we can construct a tree. So, the first one we have this sequence A, C, G, C, T, A, F, K, I. Here A is mutated to G, right, first one is A is mutated to G. So, here this A comes to G, and the second one I is mutated to L, okay, this I is mutated to L, right. So, everything is here. So, here I instead of here F, K, L. Then from this sequence, then we have next mutation alanine is mutated to glycine, right. So, here this alanine, right, this alanine is mutated to glycine. So, it starts with G. So, the sequence here also G, F, K, T. Then again A is mutated to leucine. So, here we have this A is mutated to leucine. Then go with the second one, here we have two mutations, right, one case C to S and the second one G to A. So, now we have the tree. So, you can make the alignments, right. From this tree and the number of substitutions, we will see the probability of residues to be mutated to another residue. For example, if you see A to G or G to A, as we discussed in last class, we take first sequence and second sequence, right. If A is mutated to G, for example, if you have this one, here the mutation is G to A, if this is sequence 1, this is sequence 2, this is G to A. If second one has sequence 1 and the first one has sequence 2, then the mutation is A to G, right. So, we consider these substitutions are equal in both directions, whether A to G or G to A. Okay, fine, take the element G A. So, how many frequency of pairs G A, how many mutations involved G and A?
2, 3.
1, 2, 3. So, F G A equal to 3. What is relative mutability M A? How many times A is mutated? 4, right, 1, 2, 3, 4. So, this is equal to 4, 1, 2, 3, 4. So, here normalizing factor, this is the number of mutations in the entire times multiplied by 2 times relative frequency of A residues multiplied by 100. So, this is the totally number of mutations in the entire tree. How many number of mutations? 6 mutations, right, the entire tree, right, multiplied by 2, then times relative frequency of A. What is relative frequency of A? Totally how many alanines?
Totally 10.
10 alanines, right. So, 10 A. Totally how many residues?
63.
63. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 into 7 equal to 63. So, we will get this number 190.4762. Then we get the normalized mutability, because this MA is 4. So, 4 divided by this number will give you the normalized relative mutability. So, this is 0.021. So, this here. So, now we get the mutation probability Mij, this is given as Mj into Fij divided by sigma Fij. So, here Fij, that is Mga, this is equal to 0.021 multiplied by what is Fij,
Frequency of
this is equal to 3, right, divided by sigma Fij, this is equal to 4. So, we get this Mga is equal to 0.0157. So, Fij is the total number of substitutions involving alanine, that is 4, right. So, this is equal to 0.0157. Now, we get the Rij, right, this is the value we get, logarithm of this Mij divided by Fi, this frequency of G. Mga is given as 0.157, right. So, Fg we get, this is out of 63 10 glycines. Earlier we take the value of alanine, now we take the value of glycine. So, this is equal to 0.1587, right. Then Rga we substitute the values here, log of Mij is equal to 0.0157 divided by 0.1587. So, we give the value of minus 1.005. So, you can repeat this for all the off diagonal elements. For example, if you want to get the value of RIL, what is the, how to calculate RIL? How many times I2L mutations?
1.
Only one, right. And how many I involved in this mutation?
1.
Only one, right. So, now we can calculate normalizing factor, that is the same as here, 6 into 2 multiplied by how many out of 63, how many I's?
4.
4, right. So, normalizing factor equal to 6 multiplied by 2 multiplied by 4 by 63 divided by 100. So, we get the value of 76.2. Now, we get this normalized relative mutability. So, it is only one isolation mutation, 1 by 76.2. So, this is equal to Mi equal to 0.013. Then we get this M isolation to lesion MIL, we can get these numbers, right. So, we will get 0.013 multiplied by only 1 divided by only 1. So, this is equal to 0.013. So, now we get the Rij. So, we take the logarithm of these values, finally you get this value as minus 0.685, right. You can work out in your free time. This is for the off diagonal elements. For the diagonal elements, you will get the Mjj, Mjj is here, right. For example, RAA, right, alanine will get 1 minus Mj, Mj will get from this formula and you can get these values. Then we get the values, right, Mjj, then we will get the value of Rij, right, you can get that. So, when you derive this matrix, right, so I can take a large number of data, right, in the protein sequence database, right. What is the protein sequence database?
Uniprot.
Uniprot, right. You can get the data with any sequence identity, finally you derive the matrix. This is the PAM 120 mutation matrix. So, we see this matrix, can you tell something from this matrix? There are some letters on red, right. So, all these are
Diagonal.
the same residues, there are no mutations.
No.
These residues are the highest values because the mutations, the proteins usually they do not want to mutate at certain specific positions, right. So, the same residue they like to be the same position for some functions or some stability. The next question is, for the different cases the numbers are different. For example, if you take alanine, that is 3, if you take tryptophan, the value is 12, if you take cysteine, it is 9. So, there are some amino acids which are rarely occurring amino acids. So, in these residues are very important to maintain the same position for the structure as well as function. For example, cysteine, cysteine is important for the formation of disulfide bonds. So, if you mutate the cysteine, so it has adverse effects. So, this way the rate is very high compared with other amino acids like alanine or valine. So, you can show another matrix, say PAM 250 matrix. Here also you can see the cysteine to cysteine is 12 and the tryptophan you can see it is 17, and also the same amino acids have the high values. Other than the same amino acids, if you look at the mutations, some mutations have positive values, some mutations have negative values. Can you see the positive values? For example, see here, they are in blue. So, if we see they are similar type of substitutions, right. For example, asparagine to aspartic acid or asparagine to glutamine. So, you can have the positive values, all the hydrophobic residues, right, or some small residues. Likewise, if you see some cases you have very adverse effects minus, for example, this region. For example, if you substitute serine by leucine or alanine by phenylalanine. So, if you see some mutations are acceptable by nature, some mutations are not acceptable by nature. So, based on the real frequency of substitutions, right, we derive the PAM matrix. Likewise, there is another matrix, basically Blossom matrix. If you construct the PAM matrix, if you align the make the alignments, sometimes you can see several gaps. In the case of Blossom matrix, this is also another popular matrix. Here they use only the places where they are highly conserved, where you can get the proper alignment. In this case, they avoid the regions where they have lot of gaps. So, here you can see it mainly the conserved regions. This can avoid some of the statistical problems where the substitution rate is very low for any particular pair of amino acids, that introduces some sort of bias. Like PAM matrices, so in the case of PAM matrix, lower number of PAM matrix is appropriate for comparing which type of sequences? Close related sequences. In the case of Blossom, they do the other way around. The lower number of Blossom matrices are appropriate for distant related sequences, and generally we use Blossom 62 for comparing sequences of about 62% sequence similarity. This is very commonly used in the alignment programs. Now, I show the Blossom matrix and we compare this data with the PAM matrix. If you see here, what is the value of C to C? 9, right? Tryptophan? 11. Likewise, if you have some mutations, for example, tryptophan to aspartic acid, -4. Here if you see here, tryptophan to aspartic acid, -7, right? Here there is adverse effect, here also adverse effect. So, we look into these two matrices. So, qualitatively you can see that both are similar. So, now we derive the matrices. So, what is the purpose of deriving the matrices? What is the usefulness of this matrix?
Alignment.
For alignment. For example, now we start the alignment, right? If you have two sequences, if you are not sure how to align, then we can use these matrices to compare the similar amino acid residues and score the alignment. So, I show one example. So, one example is BLAST. This is a program, this stands for Basic Local Alignment Search Tool. So, they developed an algorithm and it is also available online. So, you can use the tool to get your alignment. How it works? Let me explain this. So, first, if you have the query sequence and we have database, how to map? They divide these sequences into small bits, small bytes of length k. So, usually they use k equal to 2 or k equal to 3, right, for the case of proteins. Okay, so, now you have the query sequence Q, L, N, F, S, A, G, W. These we compare this with the database N, L, N, Y, T, P, W. So, for example, if you take into word length of 2. So, how to divide this query sequence Q, L, L, N and N, F and F, S and S, A, A, G and G, W. So, we made into overlapping segments. Then go to database, here also you made into overlapping segments N, L, L, N, N, Y, Y, T, T, P and P, W. Then where we match? First we see this Y, L, Q, L and N, L. So, we take the Q, L and N, L, what is the score? For if we take Q, L and here N, L. So, compare these values, what is Q and N? Q and N 0, right, L and L, L to L this 4, right, 4. So, total will be 4. So, if you use LN, LN, second one LN, LN, what is score for LL? LL is 4, right. How about NN? 6, N 6, 6, that is equal to 10. So, now we can use these numbers. So, we use this matrix and we make any cutoff score 8, right. This can be adjustable. So, you can put an example score of more than greater or equal to 8 and see where we have the values which are more than this greater than 8. Here if it is LN and LN, so you get 8 and NF and NY get score of 9, NF, NY what is NN? NN 6, FY, YF is 3, 3. So, 6 plus 3 equal to 9. So, wherever you get the values which are above this your threshold, then you put a star and then continue this and try to connect these dots. So, you can do this, first start with this small segment and you give the dots which is connected, and if the score is less, then it will fades away. Then you can extend it and you can get to the for the full alignment.